Hi everyone, my name is Julia Simon. I'm a junior at Stanford University, and today I'll be presenting on my research with Dr. Mick Follows at MIT, studying the metabolic demands of Riftia Pachyptila and its symbionts. So first, what is Riftia Pachyptila? That's these guys on this slide. They are commonly known as the giant tubeworm, and they can be found 5,000 feet below the surface of the Pacific Ocean along hydrothermal vents, which are among the most unique environments on the planet. That's because the water there circulates deep below the Earth's crust, is heated by magma, and dissolves lots of toxic minerals that it then brings back up to the surface and circulates with cooler water. So these environments are really dark, they're a lot warmer than the rest of the ocean, and they have higher levels of toxins that you and I would not be able to withstand. But still, the giant tubeworm, among other species, thrive there. And what's particularly remarkable about the tubeworm is that they have the fastest growth rate among marine invertebrates known to scientists, all while not having a digestive system. So scientists have been really curious to figure out why these animals are able to be so successful in such a challenging environment. Ultimately, it boils down to a symbiotic relationship they have with sulfur-oxidizing bacteria that live within a specialized organ called the trophosome. So the tubeworm takes in sulfur through its plume, circulates that along with other metabolites through its blood vessel system, and the bacteria take in the sulfur, convert it into bacterial biomass, and the tubeworm in turn digests the bacteria to build tubeworm biomass. So we know a lot about what goes in and out of the system. We know sulfur comes in and tubeworm comes out. But we don't know so much about the actual mechanism of how that's able to happen. And so my job this summer was to use models to try and describe how this system works so efficiently in such challenging and nutrient-limiting situations. So the first part of my research was to understand how the tubeworm generates energy and builds biomass by digesting its symbionts. But in order to do that, I needed to take a step closer in and look at the sulfur oxidation mechanism of the bacteria, Riftia persephone, to understand how they do the same thing, how they're able to generate energy and build biomass for the tubeworm to then consume. Lastly, I could take insights from those two parts of the system and develop relationships between the tubeworm's growth rate, metabolite uptake, digestion, along with other environmental conditions like the supply of metabolites. So the first part of my research was to derive a system of chemical reactions to describe how both of these organisms generate energy and build biomass. So this slide shows the chemicals that we know go in and out of these reactions. My job was to try and understand what goes on in those in-between steps so that we could have a better sense of the mechanism, but also be able to draw quantitative relationships between reactants and products so we can know how much sulfur goes into building bacteria and how many bacteria go into building a tubeworm. The next step was to take those relationships and insights I can get from those chemical reactions and apply them to differential equations to understand the growth rate of the tubeworm and the microbes and further understand some of the unknowns about the system that we can't understand from empirical data. So far, I've been able to knock out that first step of this project. I've derived a series of chemical reactions to describe the nutrient flow shown on this slide. And my main takeaways that I wanted to find in these models was to understand how much sulfur is needed to build a bacteria and how much bacteria are needed to build a tubeworm. So ultimately that boils down to this insight that I have on the right side of this slide. I found that about 210 moles of sulfide produce 7 moles of bacterial biomass, and those 7 moles can be converted into 1 mole of tubeworm biomass. These 3 numbers are really important because they ultimately should demonstrate the yield of both of these systems, so we understand how many metabolites need to go in to build so many units of bacteria and tubeworm respectively, and those numbers can be applied to the differential equations that I'm working on to understand growth rates and other elements of this system. So my next steps are to take those very same ratios that I showed on the last slide and apply them to these growth equations that I have on this slide. So that'll help me get at some of these unknown variables in the equation, among them being how many microbes need to be digested per year to support the tubeworm's really rapid growth rate, and how much sulfur needs to be uptaken per year or per unit time in optimal conditions so that I can compare that to data on what we know the uptake rate of sulfur is for the tubeworm to understand what the limiting factor in the system is, whether it's the tubeworm not supplying sulfur fast enough for the microbes to produce at the maximal rate, or if the bacteria are pumping it out as fast as they can. My next steps from there will be to develop even more equations to describe the relationship between metabolite uptake and other environmental limitations like the supply and availability of sulfur, oxygen, carbon dioxide, and other metabolites. Ultimately, this project has been a big challenge for me and a really exciting puzzle for me to solve in a system that I was completely unfamiliar with before this summer. But it's been an absolute blast working with Dr. Follows and learning about these incredible organisms. So I'm grateful for the opportunity to share it with you all today, and I thank you for listening.